In discussion this week, we learned that there are two primary contributors to how humans develop. We have nature, which includes our biology, our physiology, things like DNA, and we have nurture, our experiences, the culture we grow up in, the influences of our society on how we develop psychologically. We'll start this week by talking about the first, some of the biological influences. These are the influences that are going to initially shape what the nervous system looks like. So it's important to have a firm grasp of how that happens. We have to talk about DNA. And those of you who have never learned about DNA, this will be a crash course. Compared to a molecular genetics class, we will not be going as deeply into the material, but there are some basics about DNA that I think everyone should understand, so that when someone says, that is due to genes, you really need to understand what that means, the limitations, the extent to which genes or your DNA can influence how your body develops. So genetics is the science of heredity. That is the science of how, when organisms reproduce, how do you pass along the information that codes for how you should develop onto offspring, into the next generation. That is, how do we pass along those traits that are not learned through the environment, through culture? And of course, the basis of heredity is DNA, or deoxyribonucleic acid. Most of you are probably familiar with, or at least have heard of DNA. Well, this is what, at the bottom here, this is what DNA looks like. It is two strands that spiral around one another, and in between those strands, you have these connectors. And each of these connectors can be split in half such that you have two what are called nucleobases that fit into each other like a key and lock. Well, what DNA does, its purpose is that it is a blueprint. It is a blueprint that codes, that says, that contains information for how you should develop. As much as a blueprint for a building, an engineer or an architect would take out a blueprint and say, this is what the building should look like. The same is for DNA. You start with how the body, how the nervous system should develop, and then based on what you have available to you and the restrictions that are placed upon you, you can actually the end result may look very different from what your DNA has coded for. But this is where you start. To give you an idea of how this works, again, we have those two strands, and in between we have those nucleobases. Well, how does it contain information? How does it contain a code? Well, what you can do with DNA is you can rip it apart. Imagine taking those two strands, one in each hand, and then pulling on them such that you unclasp those nucleobases. You can see that here. We actually have a particular protein, a particular enzyme called RNA polymerase that is responsible for breaking apart our DNA. And once it's broken apart, the nucleobases are exposed. On the bottom left here, you can see the different kind of nucleobases. We have adenine and thymine and cytosine and guanine and uracil. I will not expect you to know that. However, you should realize that some of these go together with others. Adenine can only go together with thymine. Cytosine can only go together with guanine. And uracil is a little more flexible. But the idea is, once you've ripped apart that strand, what you can do is you can rebuild another strand by pairing together whatever the exposed nucleobases are with new nucleobases that are floating around within the cell. So you can see here what's happening is once the DNA is ripped apart, the cell is starting to create something called mRNA, or messenger RNA. And so these little pieces are going to fit into those exposed bases, and what it's going to do is it's going to read the code that is hidden within those nucleobases. If you have a sequence of adenine and then thymine and then another thymine and then a cytosine and then another adenine, what you're doing is you have a code hidden within the pattern by which those nucleobases are arranged. Messenger RNA reads that code, makes a copy of it. And once it makes a copy of it, it will then detach from the DNA as its own strand and then float out into the rest of the cell. So this is what this process looks like. If we have the DNA, it's ripped apart and then we make a copy of it. What we've done is we've made a copy of that code, and now the body can use that code to build something out of it. I will not expect you to know too many details, but here is the short of it. The body will use that code to then build protein. As you can see here, the mRNA, which is the middle image here, is made up of a copy of the DNA, and then based on series of 3 nucleobases, the body will start to chain together these chemicals called amino acids. And when you chain together different amino acids, you get a protein. That protein then does something within the body. So what DNA is a blueprint for is for protein. The proteins within your body, which control pretty much everything, are coded for by your DNA. That is what you're trying to build. And that is what sometimes can be built improperly from the blueprint. We'll get to that in a moment. Some other terms you should be familiar with. You've probably heard the term genes. Genes do not simply refer to DNA. These are different things. A gene is specifically a section of DNA that codes for a specific trait. So your DNA is very, very, very long and contains a lot of information in it. It contains really all the information that will be needed to build every part of your body. So the DNA is subdivided into different sections, and those sections are responsible for individual traits. That's what a gene is. The section of DNA that codes for a specific trait. You've also probably heard of chromosomes. What these are is they are condensed packages of DNA mixed with protein. The idea here is because DNA is so long, it's this huge strand that if it were to just free float within your cells, within the nucleus of your cells, then it would probably all get tangled up. What it wants to do is package it in a particular way such that it is manageable, it's all located in one place, and you can just kind of pull a strand off of it when you need to read it. That's what chromosomes are for. And so humans have 46 chromosomes. You get 23 from mom, 23 from dad. We'll go into some detail about that in a moment, why that's important. But here's what this strand looks like. So you have the DNA, and as that DNA is stranded throughout the nucleus, it is going to be packaged together into an entire chromosome that looks like the image on the bottom here, this kind of odd X-like shape. How is this done? How is it packaged? Well, if you take DNA, it needs something to wrap around. Imagine that DNA is like toilet paper. If you just had a bunch of toilet paper and you were trying to package it together, it really wouldn't go very well if you didn't have that little cardboard tube in the center, or if at least you didn't have that cardboard tube to start. Some toilet paper, they actually pull the cardboard out. But the idea is the same. For your cells, for DNA, the DNA will rely on something called histones. These are their own types of protein. What they are responsible for is to allow DNA to wrap around it. So you're coiling DNA around histones just like toilet paper. And when you do that enough times, what you do is you get this tightly, densely packed collection of DNA. Then when that DNA needs to be read, all that needs to happen is a little bit gets pulled off of the histone, you read that little bit, and then you put it right back. It's very neat and tidy. This is how you get a chromosome. Ooh. Okay, so why is this important? Why do you need to know that DNA codes for proteins? Well, DNA is going to be, like in every cell of your body, contained within the nucleus. And as we've learned, your neurons also have a nucleus, as you can see right here. And this DNA is going to make proteins. I said that proteins do everything, But within neurons, they have a number of very, very important functions. One, they're going to maintain and grow the neuron. Think back to when we talked about different types of glial cells, supporting cells of the neuron. A lot of these cells are responsible for maintenance and growth, but the neuron is going to maintain and grow itself as well, using its own proteins that it creates with its DNA. It's also going to determine how neurons change and develop, such as via long-term potentiation, which we've gone over plenty. At the end of the axon terminal, when long-term potentiation happens and you create more neurotransmitters and you create more of the receptors to receive those neurotransmitters, those are proteins. Both neurotransmitters and the receptors that receive them are proteins. So if you're going to change something about the neuron, you're going to need those proteins made by the DNA. So it also affects how neurons communicate through the production of neurotransmitters, which are proteins. If neurotransmitters are, if you need proteins to create neurotransmitters and they control communication, then directly DNA will control how quickly or in what way your neurons will communicate with one another. All right, well, let's give a solid example of how something within your DNA, coded within your DNA, could affect your nervous system, and therefore everything we've been talking about in terms of learning, memory, your conscious experience, etc. My argument here is that small changes in DNA can actually have very large psychological consequences. And the example that I want to use here is sickle cell anemia. Some of you may have heard of this. This is a genetic disorder that is very common within African populations. The idea here is there is a single mutation caused within the DNA that causes red blood cells to look a little different. So normally, if you see this image here, normally your red blood cells look like they do on the left side. They're kind of large, red, puffy, kind of happy. These are soft, they're pliable, and as they're running through through your veins, through your arteries, if they kind of bump up against one another, they're very soft and pliable, so they just kind of pass on by. You're not prone to clots. Versus if you have sickle cell anemia, either some of or a vast majority of your red blood cells will look like what they do on the right side here, kind of deflated looking. But more than deflated, what these actually are, they are hard and they're crystalline. So rather than being soft and pliable, they're very hard. And so as they pass by one another, they are prone to clotting. They're prone to grouping together. More than that, though, what normal red blood cells have over sickle cells is that they can more easily carry oxygen, which is what their responsibility is. Blood transports oxygen throughout your body. Sickle cells happen to not be able to do that very well. So not only are they prone to clotting, they don't carry as much oxygen. If they don't carry as much oxygen, that means that your muscles, the rest of your body, is not getting what it needs to produce energy. Your body relies on oxygen to produce energy. So without it, that can actually lead to quicker exhaustion. Well, think about this. If you are someone who has this genetic disorder and your blood cells do not carry as much oxygen, and therefore your muscles and whatnot may not produce as much or be able to use as much energy as they otherwise would, how would that affect your life? Well, if you are not getting enough energy and you are chronically exhausted, might that affect how well you pay attention in class? Certainly when anyone is tired, they have a harder time paying attention, of staying awake in class. Or how often you hang out with friends. When you're tired, that is the moment where you're saying to yourself, do I feel like going out tonight? Or do I feel like just kind of staying in and doing my own thing? I'm pretty tired. I think I'll stay in. So to some degree, this could make you less likely to hang out with people, to want to do things that are more active. And how could it affect your personality? Certainly, when we're more tired, you could be grumpier, you could have a shorter fuse with someone. This is just one example, but there are many types of these kinds of disorders where it takes just a very small change in the DNA to cause something that can have very large, what are called downstream consequences. That is, if you take this kind of, if you look at how, say, changes to this very basic protein can have effects on much larger psychological traits, it's really quite astounding. And I encourage you to think about genes in this way. For the most part, changes in the genes will not produce these very large effects, but sometimes it can. And even if it produces a very small effect, these effects can add up over time to create much larger effects or consequences for your psychology. Okay. What we'll get into next time is, now that we have a basic understanding of how DNA can shape our psychology, we'll next get into genetic inheritance. That is, how do we take DNA and pass it on to our children?